Come to my age once, then you will see how it is. Ever heard? Adults from 40, 50, and older use this or similar sentences to justify their state of physical and mental decline. Let me tell you one thing. The state that many people are in after 30 is not normal. You are not a victim of your genes. You can look very good, stay fit and healthy well into old age if you do the right things. And most importantly, if you don't accept that this prevailing decline in people is normal. Don't let those old folks talk you into it. Personally, I never accepted it. Even as a small child, I intuitively knew that those sentences simply couldn't be true. Back then, I had no clue about health and genes, but I never wanted to believe it. It even got to a point where I argued with adults. Of course, a little boy with no life experience didn't stand a chance, only they had life experience. In this video, I will explain to you exactly why you are not a victim of your genes and what you can do to stay forever young. But first, follow for more. Epigenetics is the part of biology that studies how environmental factors influence the activity of our genes without changing the actual DNA sequence. It is the central control center of gene regulation, the conductor of a genetic orchestra. DNA methylation. A small chemical group called the methyl group is attached to certain building blocks of DNA. This mark often acts like a gene off switch, meaning the affected gene is read less or not at all. DNA methylation is crucial for each type of cell to fulfill its special role. For example, it ensures that a skin cell uses different genes than a liver cell, even though both have the same DNA. Histone modifications. Our DNA isn't naked in the cell nucleus. It is wrapped around special proteins called histones, like a thread around the spool. These histones can be chemically modified, for example, by adding acetyl groups, which activate genes, or methyl groups, which can switch genes on or off, depending on the site. Such changes affect how loosely or tightly the DNA is packed, and therefore whether a gene can even be read. This is called the histone code, which acts like extra information deciding which genes are active. Non-coding RNAs. Besides DNA, there are special RNA molecules that don't encode proteins but still play important roles. These include, for example, microRNAs or long non-coding RNAs. These molecules act like small directors, deciding whether a genetic message, mRNA, is translated into a protein or immediately degraded. They thus provide a fine-tuned control of gene activity, often in a targeted and fast manner. Genetics versus epigenetics. While genetics represents the fixed blueprint of our organisms, epigenetics determines if, when, and how this blueprint is used. Comparable to a piece of music, the notes, genes, remain the same, but the interpretation, epigenetics, decides on tempo, dynamics, and expression. Environmental factors in detail. How our surroundings influence our genes. Let's start with nutrition. Food is a genetic switch. Nutrition directly influences epigenetic processes via the one-carbon metabolism, which supplies essential methyl groups for DNA methylation, like B vitamins, B2, B6, B12, folic acid, choline, methionine, as adenosylmethionine as the central methyl group donor. Here is a study for a human example. Maternal undernutrition in the second trimester led to lifelong hypomethylation of IGF-2, a growth-associated gene. Consequences: higher risk for obesity, coronary heart disease, glucose intolerance. Environmental toxins and stress, how our environment affects genes. Examples: tobacco smoke turns off the AHRR gene, a marker of smoking. Consequence: higher cancer and inflammation risk. Alters methylation of inflammatory genes, e.g., IL-6, TNF alpha. Increases risk of asthma, heart attack, and stroke. Next, pesticides and plasticizers disrupt epigenetics of fertility, brain, and immune system via germ cells, often across generations. Chronic stress and trauma. What is important to know here, not every strain equals chronic stress. If you can cope with stressors, there is no stress. I will make a whole video about it. Want to know how real stress occurs? Write it in the comments. Next, NR3C1 gene becomes more methylated, leads to less stress regulation, more cortisol. FKBP5 disrupts the HPA axis, higher risk of depression and PTSD. Meditation lowers FKBP5 methylation and inflammation after just 8 hours. Metabolism and type 2 diabetes. Genes for fat burning and appetite. PGC1 alpha, CPT1A, FTO are epigenetically altered. Consequences: insulin resistance, overweight, inflammation. Brain and psyche. Diseases such as Alzheimer's, depression, or schizophrenia show typical epigenetic patterns in the brain. Environment, diet, and toxins regulate neural connectivity via histone changes and miRNAs. Epigenetic inheritance. Not only genes, but also epigenetic markings are inherited. Example: pesticides in great-grandparents leads to fertility issues in great-grandchildren, even though they were never directly exposed. Okay, what can we do about it then? How can we consciously influence our genes? Exercise and sports lead to demethylation of PGC1 alpha, leads to more mitochondria, which leads to metabolic resilience, effects on muscle plasticity, insulin sensitivity, inflammation, regulation. Meditation, mindfulness, and yoga. Stress relief leads to positive epigenetic effects on FKBP5, NR3C1, reduction of systematic inflammation markers. Nutrition, rich in plant-based, unprocessed, organically grown foods. And don't worry, there is also room for animal products, but 80 to 90% plants. And only if the quality is right. Don't be cheap, buy quality. You don't know what that means? Watch my food quality video right after this one. You will learn everything. This influences histone modification, DNA methylation, antioxidant gene signaling, sleep and circadian rhythm. Sleep deprivation changes expression of over 700 genes, especially those related to immunity, inflammation, and cell repair. Methylation of clock genes, e.g., PER1, BMAL1, affects the internal clock. Sleep deprivation leads to hypomethylation of metabolic genes in fat cells, which leads to increased insulin resistance. Light and vitamin D. UVB radiation stimulates vitamin D production, which leads to vitamin D acts as an epigenetic modulator via the vitamin D receptor, influences genes for immune function, cancer defense, bone metabolism through histone acetylation. Vitamin D regulates over 900 genes via epigenetic mechanisms. Gut microbiome. Microbes produce short-chain fatty acids like butyrate, which inhibit histone deacetylases, HDACs, promotes anti-inflammatory gene expression. A dysbiosis equals epigenetically mediated inflammation and metabolic disorders. Diet influences the epigenetic structure of peripheral tissues via the microbiome, even in liver and brain. Thoughts, emotions, and genes. Mind influences genetics. One of the most revolutionary findings in modern biomedicine is psychosomatic epigenetics, meaning our thoughts, emotions, and inner beliefs measurably influence our gene activity. Chronic stress leads to epigenetic alarm readiness. Mental chronic burden, e.g., worry rumination, activates the HPA axis, which leads to cortisol release. Cortisol permanently changes methylation of stress sensitive genes like NR3C1, FKBP5, increasing vulnerability to depression, anxiety, and autoimmune diseases. Positive emotions, meaning, and mental strength lead to genetic resilience. Meditation, gratitude, optimism correlate with increased expression of health promoting genes. People with high life satisfaction showed reduced expression of inflammatory genes and activation of antiviral gene programs. Top down regulation. The brain as the epigenetic control center. The brain influences epigenetic patterns in other tissues via neural, hormonal, and immune signaling cascades. Mind to nerves to hormones to epigenetic modification. This psychogenetics is an active field with relevance to psychosomatic medicine, placebo effects, psychoneuroimmunology. So, what have we learned? Basically, our environment writes in our gene book. The idea that our genes are an inevitable fate is BS. What crazy person even introduced a crazy idea in the first place? Crazy. But honestly, if a little shit like me saw through all that bullshit back then, how could this have remained accepted as normal for so long? Someone must have had interest. But let me shut up right here. Make your own conspiracy theories. Epigenetics shows that genes are like tools. Their use depends on our environment, our experiences, and lifestyle choices. Who we are, how healthy we age, and what we pass on to the next generation depends on how we live. Oh, heavy stuff. That was a lot, but worth it. I think I changed your life again. My pleasure. What? You are addicted now and want more? No problem. Like I told you, next is chronic stress. So stay tuned. And now, follow for more.